Molecular docking and scoring is one of the very important topic in bioinformatics. One of the very important procedure in computer-aided drug design. So we already completed the practical aspects of molecular docking. So we did rigid docking as well as the flexible docking, right? So we'll see the theoretical aspects of molecular docking and scoring function. Sorry. So many times already I defined what is meant by molecular docking. Even if you look at the videos I gave for practical session, I gave some theoretical background for molecular docking. So in simple way, we can define molecular docking as a procedure for making complex between two molecules. We can also make complex between more than two molecules, but mostly we make the complex between two molecules. And in terms of drug discovery, we we make the complex between a macromolecule and a small molecule. So this macromolecule, it can be either a protein molecule or DNA or RNA, and the small molecule is the drug molecule. So writing some computer algorithm to make possible complexes between the given protein molecule and the drug-like molecule. Those algorithms are what are called as the molecular docking algorithms. So what we actually evaluate here is that what are the possible interactions which can take place between the protein molecule and the ligand molecule. That is what actually we evaluate. That is why in the previous lecture I discussed about the various possible interactions between the drug molecule and the drug target. So using the mathematical computer algorithm, so we evaluate that how we can fit the drug molecule inside the given binding pocket for the given protein molecule. What will be the energetics, what type of interactions possible between the drug molecule and the drug target, right? So what actually we do in molecular docking is that we predict the structure of the intermolecular complex formed between the two or more molecule, most often two molecules between the protein molecule and the ligand molecule. So what we can expect out of the docking algorithm is that docking algorithm will suggest what's the preferred binding mode of the ligand molecule or the drug molecule. So because the same drug molecule or the ligand molecule can be fitted in different conformations or different orientations within the binding site. This docking algorithm will tell that which orientation is the best possible orientation to fit the ligand inside the binding site. So for this, since different ways we can fit the ligand molecule inside the binding site, there should be some scoring function to score that which orientation is the best possible orientation that is having a good energetics. So that is why where we have the scoring function, we will see that later, right? So docking problem is that even with a lot of advancements, right? So still we have some limitations in any docking algorithm. So basically we can classify this docking algorithm into two types. One is called as a rigid body docking, another one is the flexible docking. Again I repeat, broadly we can classify the docking algorithms into two types. One is the rigid body docking, another one is the flexible docking. So if you take any docking algorithm, there will be two components in it. One is called as search algorithm, another one is the scoring. Again I repeat, broadly we can classify this docking algorithms into two types. The first one is the rigid body docking, another one is the flexible docking. So rigid body docking is the docking in which we consider the protein molecule and the ligand molecule as completely rigid. So flexible docking is the thing in which we can give flexibility for either for the protein molecule or for the ligand molecule or for the both. That is what is called as the flexible docking. So we have used okay, we completed two exercises, right? So can you tell the first one is a kind of rigid docking or flexible docking? Is it a rigid body docking or flexible docking? I mean the second exercise, what you have done.
Rigid docking, sir.
Okay, so practically speaking, that is not rigid docking. We named it as a rigid docking because we consider the protein molecule as a rigid body. I'm talking about the second exercise, what you did, the practical session. We named it as rigid docking, but actually it is not rigid docking, right? So have you given any flexibility for ligand molecule or not in the second exercise? I hope that you set the torsional degrees of freedom. How many number of rotatable bonds there in the ligand molecule? So how many bonds you kept as rotatable for the ligand in the second exercise?
6, sir.
6 means, right? So even if you give if you give one bond as a rotatable bond, we cannot say it as a rigid body docking. Are you able to understand? Rigid
Yes,
body docking has zero rotatable bond in protein and zero rotatable bond in the ligand. Uh, don't get confused, right? So we name it as rigid docking, but practically speaking, that is not rigid docking because we gave some flexibility for the ligand molecule. That is also comes under the flexible docking. So what is a rigid docking? There is no conformational flexibility either for the protein molecule or for the ligand molecule. Okay, just like the lock and key model, right? So as I said, that any docking algorithm, irrespective of whether it is a commercial software or it's a free software like Autodoc, doesn't matter, right? They'll be having only two component. One is the search component, another one is the scoring component. Search component is used for finding out which way we can fit the ligand molecule inside the binding site, and scoring component is what is the score for that specific conformation. So these are the major two components of any docking algorithm. So coming to the docking problem, so it involves many degrees of freedom. So generally speaking, at least 6 degrees of freedoms we have to consider. 3 degrees of freedom corresponding for the translation and 3 degrees of freedom for the rotation. So if we take the protein molecule, we can move the protein molecule in XYZ direction. We can move the ligand molecule in XYZ direction. So that is what is called as the translation. 3 degrees of translation, X direction, Y direction, and Z direction. So similarly, we can rotate the protein molecule in 3 directions. We can rotate the protein in X direction. We can rotate the protein in Y direction. We can rotate the protein in Z direction. Similarly, we can rotate the ligand molecule in X direction, Y direction, and Z direction. Okay, so these are called as the 6 degrees of freedom. Even if you write a rigid body docking algorithm in which there is no flexibility for either the protein molecule or the ligand molecule, you have to consider the 6 degrees of freedom. So take the protein molecule, move the protein molecule in XYZ direction, take the ligand molecule, keep the ligand molecule inside the binding site and move the ligand in XYZ direction, and rotate the protein molecule in XYZ direction or rotate the ligand molecule in XYZ direction. The ultimate aim of doing this, considering this 6 degrees of freedom, is that we have to fit the ligand molecule in proper orientation inside the binding site. Is that understandable?
yes, sir.
So those are the 6 degrees of freedom. But apart from that, we have another one important type of degree of freedom called as the conformational degree of freedom. So the conformational degree of freedom comes into picture if the algorithm is a flexible docking. For rigid docking, there is no point of talking about conformational degrees of freedom. But if it is a flexible docking, then we have to always talk about consider the conformational degrees of freedom. So this conformational degrees of freedom is applicable for both the protein molecule and the ligand molecule. Let's take ligand molecule. So if you take any small drug-like molecule, it may contain at least a few numbers of carbon-carbon single bond. Those bonds are rotatable bonds. So we can rotate the bond. So if we rotate the bond, carbon-carbon single bond, wherever it is there, right? So then the molecule can take different conformation. So now you have to think of 3 translational degrees of freedom, simultaneously 3 rotational degrees of freedom, simultaneously n number of conformational degrees of freedom. So the complexity of the algorithm now get increased. Translation, rotation, along we have to account for the conformational degrees of freedom. So that is why flexible docking algorithms are kind of advanced algorithm. Even if you give the flexibility for the entire ligand molecule, so ligand molecules will be having very few number of carbon-carbon single bond. We can easily manage the three rotational, translational, along with many number of conformational degrees of freedom for ligand. But if you think of giving conformational degree of freedom for the entire protein molecule, it will be very difficult to manage, because even if you take protein constraint, if you consider protein molecule made up of 200 amino acid, right, there is going to be lot of carbon carbon single bond. If you make all the carbon carbon single bond rotatable for the protein molecule, the number of conformations going to be exponential. So the computer algorithm may not able to manage that. That is what we call it as the computationally expensive procedure. So even the flexible docking algorithm, they can allow you to give flexibility only for few number of amino acids which are present within the binding site, not for the entire protein molecule. If you give flexibility for the entire protein molecule, maybe your computer resource may not be enough, or it may take several years for you to solve that problem. Is it understandable? This is what is called as the docking problem. Is that understandable? Hope I am audible.
Yes, sir.
Okay, let me summarize that. Rigid body docking is three translational, three rotational degrees of freedom for the protein molecule and the ligand molecule. Flexibility docking is 3 degrees of freedom for rotation, 3 for translation, along with that many number of conformational flexibility. Okay, so another thing is that we can classify, another way to classify the docking algorithm is that we can classify the docking algorithm into automatic and manual. So automatic is very simple. You have to probably in the next, next exercise you'll be doing kind of automatic, right? So we won't set any parameter, just input the protein molecule and the ligand molecule. So all other parameters will be taken care by the algorithm itself. So autodoc, which comes under semi-automatic manual. If you are a very experienced person, you can control various parameters in autodoc. So there are some algorithms which are considered as manual algorithm, in which the user has to set all the parameters, right? So that's another way to classify the molecular docking algorithm. So here I have listed the various very commonly used, the most important docking and scoring algorithms which are available for protein ligand docking. So DOCK, UCSF DOCK, University of California, they have written the algorithm. UCSF DOCK, very good program. Autodoc we have used, right? So Scripps Laboratory, they have written the algorithm. FlexX, we have a license for FlexX. It's a commercial software. Glide is a part of Schrodinger. Again, it's a commercial software. Gold is also commercial software, and many more, right? So there are hundreds and thousands of docking algorithms which are available. Just for understanding, I have given few algorithms, right? So here few algorithms are like rigid body docking. We can use it for rigid body docking, and few we can use it for flexible docking. So if you take autodock, we can either go for rigid docking or flexible docking. If you go for dock, we can go only for the rigid docking, not for flexible docking. FlexX is very good for flexible docking, and so on, right? So depending upon what you needed, right? So you have to choose the docking algorithm. For every problem, all the algorithms will not be good. So certain problems we have to choose certain specific docking algorithms. So what I'm going to do is, in the next class, I'll be giving some, overall idea about what are different search parameters which are available. So in this, I am going to focus only on the first algorithm called as dock algorithm, right? So as I said, dock is a program, docking program written by University of California, UCSF dock, the name itself. It's a very good algorithm. It comes under rigid body docking algorithm. So which means that we cannot give flexibility for the protein molecule or the ligand molecule. We have to keep both the protein molecule and ligand molecule as a rigid component. So this is an example for rigid body algorithm, docking algorithm. So I'm going to explain you what are the various steps of this rigid body docking algorithm. Because the complexity of flexible docking algorithm is more. That's why we are seeing the simple docking algorithm, right? The very first step, docking, comes under structure based drug design. We know that for any structure based drug designing approach in computer aided drug design, we should know the 3D structure of the drug target. So one dedicated lecture I discussed about what's the role of X-ray crystallography in structure-based drug design. So even though we have many methods for solving the 3D structure, always the drug designer prefer for a X-ray crystallography structure because they believe that it can trap the biologically active conformation of the protein molecule. The another advantage is that we can co-crystallize the drug molecule or ligand molecule along with the protein, and we can crystallize along with the protein with ligand or the drug molecule. That will tell where the binding site is present, what type of interaction is made between the known ligand or the drug molecule, potential inhibitor with the drug target. So same thing here. The very first step is we should have the structure, 3D structure of the drug target. Most preferred way to get the 3D structure is the X-ray crystallography. So while choosing the structure for molecular docking, we have to ensure that the structure that we take from protein data bank is of very less error, very minimum error or no error. So when we can say that the structure that we choose is of good quality, so the various parameters which are related to X-ray crystallography, that is going to determine the quality of the structure. The parameters like resolution, it plays an important role. So normally, if the resolution goes beyond 2.5 or 3, definitely, we won't take that. We won't consider that structure for molecular docking. The reason is that the diffraction pattern or the electron density map of those structure will not be good. So there may be some error because of that while fitting the amino acids to the structure. But if any structure that is available at the atomic resolution, like in the range of 1, 1.5, that is very good. So we have to look for the structure which is having good resolution. R factors, we have to look, that how well, uh, how accurately they fitted the amino acid. That is dictated by another value called as R factor. Like that, we have different parameters should be considered while choosing the structure for molecular docking. Most preferably, we should go for the structure which is co-crystallized with the drug molecule or ligand molecule. But even in the absence, we can some docking algorithms, it can find where the binding site is present. And UCSF DOCK is one such algorithm which to which if you input the protein, it itself will find out where the binding site is present. So the first step is we have to get the 3D structure through X-ray crystallography or any other method. Most preferred is the X-ray crystallography. The second step is that we have to find out or we have to define the molecular surface of the binding site. Computationally, this is the first step. Okay, computationally, the first step. Once if you input the protein molecule, your algorithm should be able to tell that where the binding site is present. That is the second step or first step. If we consider computationally, this is the first step, right? So for that, normally what normally all the algorithm or UCSF what it does is like it identify each atom in the binding site and it calculate what is the van der Waals radius of each atom. So here you can see that these are the various atoms of the amino acid, and it calculated what is the van der Waals radius of each atom. So each atom here is represented by its van der Waals radius. Okay, that's a very simple thing, right? Um, so once if you represent the atoms as the van der Waals radius, so it will be easy for you to find out where the binding site is present. There are different algorithms which are available for finding the binding site. One simple algorithm or very frequently used algorithm by many researchers is that using the solvent accessible area calculation. Okay, so it's a very simple thing. So here, this part is the same thing what I have shown in my previous slide. Van der Waals radius of each atoms which are lining the binding site. So to find out the exact volume or the area of the binding site, so what we have to do is that we have to use something called as probe. So probe is a kind of atom, chemical atom, so with some radius. So normally, the most commonly used probe atom is the water molecule, H2O. So we know what is the van der Waals radius of H2O. So this blue color, okay, which represents the van der Waals radius of the water molecule. So they will roll this water molecule over the van der Waals radius of the atoms which are lining the binding site or the surface. So what will happen if you start rolling the water molecule, right? So this water probe will make contact with different part of the van der Waals radius of the atoms lining the binding site. So it will roll. Literally speaking, it will roll and make contact in certain position of the atoms which are represented as van der Waals radii. So here, if you look at this is the path, okay, which is traced as where the water molecule made contact with the van der Waals radii of atoms lining the binding site. Okay, so this is called as a re-entrant. This is con- convex surface. Okay, I hope you can understand why it is called as convex surface, because the shape is like convex. So this region is what is called as the re-entrant. So what's the meaning of re-entrant? The solvent water molecule, the probe atom, can maximum reach up to this, and it cannot go beyond that point. This is maximum contact, maximum region to which it can penetrate. Beyond that, it cannot penetrate. So that's why this region is called as a re-entrant. It's the maximum it can enter. Even though you have the cavity here, you have the hole or vacuum or space, whatever way we say, right? So this is a maximum that water probe can contact. That's why it is called as the re-entrant. So coming to the characteristics of probe sphere, water molecule, right? So the probe will be having the dimension of 1.4 to 1.5 angstrom sphere. That actually represents the radius of the water molecule. Okay, uh, that is how the binding site can be identified. Is that understandable?
Yes, sir.
So step 2 is we are finding the binding site, right? So we are using the solvent accessible area. So the third step is we have to position the ligand within the binding site. That's the third step. So for positioning the ligand molecule inside the binding site, here we know, if we see this image, we know that this is a re-entrant. Okay, this is a maximum region where the probe can contact. All these things are fine, right? But the thing is that how this information is understandable by the computer. Okay, we have to make some way that the computer can understand that where the binding site is present. So for that, what normally they do is that the empty space of the binding site, the empty space here is the solvent accessible area. So wherever the solvent can go and get in contact, that region is called as the empty space of the binding site or the solvent accessible area. So the empty space of the binding site has to be defined way that computer program can understand before ligand can be inserted. So which means that, so we have to define to the computer that this is the place where the binding site is present. This is the shape of the binding site. This is the volume of the binding site. This is the size of the binding site. That first we have to define to the computer algorithm in a very understandable way for the computer, right? So for that, what they do is that, so once if they know which area is the solvent accessible area, so here if you take, definitely this region is the solvent accessible area, because once if you roll the probe, right, so that it can easily make contact with those region, right? So based on that, we can say that this is a place where the binding site is present. Okay, how to make this space as the binding site, which can be understood by the computer algorithm. So they will fill this space by using sphere of different sizes. There's a condition used in UCSF uh, uh, DOCK. The condition is that, so whatever sphere you're fitting inside this should make contact in two position. It should make exactly contact two position of the binding side. So if we take this sphere, so it is making contact in this position and this position. Similarly, if you take any sphere, if you take this sphere, it is making contact in this position and this position. If you take this sphere, smaller one, it is making contact in this position and this position. So similarly, you have to fill with many number of sphere, but the condition is that each sphere within the binding site, okay, it occupies within the binding site, should be in contact with the binding site in two places or two positions. Is that understandable?
Yes, sir.
So now we know that these are the wherever we have the sphere, right? So that is the binding site. So we created some negative image. Okay, so now we can easily tell that this is the binding site. Wherever we have the sphere, that's the binding site. That is the place where the ligand molecule should be kept inside. Okay, so that is what we have to do. So the empty space of the binding site is defined by identifying a collection of different sized sphere which will fill up the space available and give you a negative image of the binding site. This is what I explained, right? So we created the negative image. Now this information is more than sufficient for the computer to understand where the binding site is present, right? So fourth step is, like, we have to orient the ligand molecule inside the binding site. So this is they will look for the shape similarity, different algorithms there. I'll be discussing the next class, right? uh, uh, Shape similarity algorithm is the commonly one that is used for fitting the ligand molecule inside the binding site. So one thing is that, if you look at the number of sphere in this image is larger, more number of sphere, but the number of image is reduced here. Okay, so in this step, moving from third step to fourth step, so some number of spheres will be merged together to reduce the number of sphere. Again, I repeat. So third to fourth step, some number of spheres will be reduced wherever you have the overlap, right? So those spheres will be reduced to one sphere. That's why the number of sphere is reduced here. So they have labeled the sphere completely. It is filling the binding pocket. So they have labeled the sphere. Centroid of the sphere is A, B, C, D, E, F, G, so on, right? Similarly, if you take any ligand molecule, the ligand molecule is made up of atoms. So they will convert the each atom as a pseudo atom. So here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, this number, this actually dictate the pseudo atom. Okay, so now this is again made up of sphere. The binding site is made up of sphere. So we have to align the ligand molecule to the spheres which are present in the binding site. So in which what they will try is that the centroid of most of the pseudo atoms in the ligand molecule should be overlapped with the centroid of the spheres which are present in the binding site. Again, I repeat, the centroid of the pseudo atom of the ligand molecule, maximum number should be matched with centroid of the spheres which are present in the binding site. That's why it is called as matching the shape. Is that understandable?
Yes, sir.
Okay, so that is what they do. So this procedure, we can fit the ligand molecule in different orientation. Okay, there may be more than one way we can fit the ligand molecule inside the binding site. In the sense, more than one way we can fit the pseudo atom centroid and the centroid of the spheres which are present in the binding site. So here we can say there is a kind of good shape complementary. The top image, it shows that there is a kind of good shape complementary between the pseudo atoms and the binding site. But if you look at the same molecule oriented in different way, there's a kind of steric clash. the pseudo atom 11 is making some kind of steric clash with the binding site. So this way we can say that some orientations are preferred orientation for the ligand molecule within the given binding site, and some orientations are not preferred one. Is that understandable?
Yes, sir.
Okay, so this is how the alignment is made between the drug molecule and the binding site in case of the DOCK algorithm, right? Uh, the concept will be very different for different algorithm. This is only applicable for the rigid body docking UCSF DOCK algorithm, right? So since we can fit the ligand molecule in different orientation within the binding site, okay, which orientation is the best one? That's the next question. All right. So far, the first component of the docking algorithm is over. That is the search algorithm. The search algorithm is trying to search which conformation of the drug molecule can be fitted well inside the binding site. But at the same time, there may be more than one way by which we can fit the ligand molecule inside the binding site. If we can fit more than one way, so which one is having the best complement? That is the one we had addressed. That is addressed by using the scoring function. Second part of the docking algorithm. All the docking algorithm will be having this scoring function for evaluating the score for the fitness score, right? This is a simple way to calculate the binding energy, right? So some algorithms may use very complicated scoring function. So what I'm showing here is the very simple way to understand what exactly is the meaning of scoring function, how what it actually calculates. It actually calculates the binding energy. So if you remember in the previous lecture, I not in the previous lecture, in the laboratory session, I have told what is meant by binding energy. So binding energy is the amount of energy that is released because of the interaction of the ligand molecule with the binding pocket amino acid. So mathematically, we can say that it's going to be contribution of different type of energy. So simple formula for calculating this delta G bind is delta G solvent, the solvation energy, delta G conf is the conformational energy, delta G int is the interaction energy, delta G ROT is the rotational energy, delta G TR is the translational rotational energy, delta G BIB is the vibrational energy. So like this is a very simple mathematical expression for finding the delta G binding. It is approximated one, right? So we can add many more things to make this more realistic than a simple one like this, right? So this is the one which is implemented in the Autodoc, right? So what is the scoring function? How they calculate the score? So this is a formula that is used in Autodoc. Delta G is equal to uh, V bound minus unbound plus V bound minus unbound plus bound minus unbound plus delta G delta S conf, right? So here the L which represents the ligand molecule and P which represents the protein molecule in a ligand protein docking calculation. So it calculates the pairwise energetic terms. Okay, so there's a lot of expression. What is V here? So this is an expression for V. You can see how complicated it is, right? So as I said that the scoring function may be even more complicated if you go for real flexible docking algorithms like FlexX, or if you go for Glide. Okay, so these are the mathematical expressions that are used to find out that which of the fitted conformation is the best conformation. That is again calculated with the help of the energetics, right? So that is something about docking and scoring function that is used in dock.